So we're now recording so that we've got all of this for everybody to look at. Has anybody gone and looked at made sure you can like see the YouTube videos and those things and actually gone and look at any of that yet? No? Okay. YouTube tells me nobody has, or at least nobody's tried to watch one of the videos. I just didn't know if making sure anybody had kind of gone and checked those things out. So they are there if you need it. So all right, so we'll wait a minute or so, let you guys do some math. Okay. So let's start with this. All right. I'm just going to randomly sort of pick someone here. Let's do this. Ireland. You're the lucky or unlucky person. How many minutes of physical activity did you get yesterday? Like just in sum total, how many did you get?
About 50 minutes.
About 50. Okay. Of that 50, how many minutes were what you would think of as being moderate versus how many were what you were thinking of as being vigorous?
I would say about 35 moderate and about 16-ish minutes vigorous.
Okay. 16-ish vigorous. That's a very kind of interesting.
Well, I ran on a 2-mile run on the treadmill, so it was exactly 16,
okay.
27.
We know that Ireland was running at 8-minute mile pace for 2 miles on the treadmill. Okay. Very good. Would you mind like sharing any of the things that you did that you thought were moderate?
I went grocery shopping.
Okay.
And then I guess carrying them up 4 flights of stairs twice.
Okay.
And
That might almost be vigorous, but okay. Yeah. Good. Okay. Excellent. So some grocery shopping. And that was all that you did the rest of the day. You did nothing else where you like stood up or walked around or anything. You just like sat on the couch the rest of the time?
I went downstairs and laid out by the pool, made dinner. That was really it.
Okay. Probably laying out by the pool, not exactly physical activity. But getting there, maybe so. Okay. Good to know. Excellent. Let me pick someone else. Taylor, you're sort of next to Ireland on my screen. Taylor, how many minutes of activity do you think you got approximately yesterday?
195.
195.
I think.
You think, okay. Of that 195, so we're at like 3 hours and 15 minutes or something. Of all of that, how much was moderate? How much was vigorous?
120 was vigorous and 75 was moderate.
Oh, okay. What were you doing for 2 hours, if you don't mind me asking, that was vigorous?
Basketball practice.
Basketball practice. Okay. And so for the entirety of the 2 hours, just like straight, you were moving at a very vigorous intensity the whole time, or are there like stops and starts in basketball practice?
There really wasn't any stops or starts. It was all going.
All going. Okay. Well, that's a lot. Congratulations on doubling your physical activity recommendations for the week in one day from a vigorous intensity standpoint. And so then your moderate stuff, like what do you think you were getting from the moderate standpoint? Like what were you doing?
I was just shooting, like just getting shots up.
Okay. I joke with my daughter. She has a little basketball goal that we let her get some shots up a lot. She likes to dunk and she likes to watch me shoot. But I've got her to say, get buckets. And she really seems to, she thinks that's pretty funny. So, okay. So basketball practice. That's probably right. We would think about that as maybe exercise in some ways, or maybe not, but we're working on skills and those kinds of things. So, okay. Anybody put like other much more mundane things on their list, like showering and brushing your teeth, or just kind of really silly things like I cleaned my apartment or my house, or I was vacuuming, or I was doing yard work. Okay. Gabby and Addie, you guys put those kinds of things. Addie, what were you doing that were kind of those mundane-ish things?
I just put like I vacuumed and like I wiped down all the counters in my house and stuff. And then
Okay. So do we think that's light intensity or moderate intensity? I guess it might depend upon how vigorously you were sort of wiping stuff down.
I just put like low intensity. Like I have hardwood floor, so it's not like hard to push the vacuum or anything.
Okay. Excellent. Gabby, similar for you? I know you had put your hand up that you had done some of those things.
Yeah, I put doing laundry and then like putting the clothes away and then like putting sheets on the bed. But all that is like light.
Probably light. I'm not going to ask you if you had to try to fold a fitted sheet, which is like maybe the single most difficult thing that being a sort of functioning adult requires. Who cares about everything else? It's those damn fitted sheets.
Yeah, it's better than trying to put a duvet on, like a duvet.
Oh yes,
insert.
we have one of those too. And my wife, every time she wants to wash the duvet, I'm just like, no. Let's not do this. It's not worth it.
Yeah, if I had to do that, that would for sure be vigorous. I work up a sweat every time I do that. So, but didn't have to do that this time.
Okay. Very good. So the point of this exercise, I hope, okay, is I wanted you guys to begin to understand and begin to pay attention to all of the things that you do every day that would be considered physical activity. All right. So now what I want you to do is think about what you're doing now. You have class, right? We're having class virtually. Imagine what you would be doing if we had class on campus. Okay. We were in Norman, we were on campus. Think about where you have to park. Think about how you get to campus. Think about how much you walk around, okay, in order to go from one class to the other, right? Our building is down on the south side of campus. If you've got other classes that are up more so on the north side of campus, if you've got to walk, right? And then you've got 12 minutes or whatever it is in between classes, because when you have exercise phys, when Dr. Mike keeps you past the time that it ends, because he's going to continue lecturing, you've got to sprint up to get to physics or chemistry or I don't know, history of something else, right? Think about how different, right, things are because of what you're doing right now. Okay. I also want you to think about how different things are. Think about what, let's just say, maybe your parents are doing. Okay. Think about what they do in their everyday life. Those of you that have maybe moved back home and you're living at home and maybe you see your parents every day and all of this, think about what they're doing. Okay. And I can tell you kind of what it's like at the Black household in some ways. Having a toddler, you're much more physically active because you have to chase her ass around all the time. But, like we get up in the morning and we get up, my wife takes a shower, she gets ready, she goes to work and she sits at her desk most of the day. She comes home for lunch and she stands up and she has a sandwich or something. And then she goes back and she sits down all day. Then she comes back home and we chase Ellis around and we're pretty active. Right. But think about how different your sort of day is, right? If you've got a desk job. But then also think about, let's just say that you have a job that doesn't involve having to sit at a desk all day. You've got a job where maybe you have to travel around and go check on people or see on people, or you have a job that requires more sort of what we would think of as kind of manual or physical labor, right? You're someone that is a plumber, or like the guys that are putting up the fence at our house, or you're a painter, all of these other kinds of things, which I'm just thinking of stuff that's going on, right? But your job is going to, by necessity, right, give you lots and lots of physical activity. Okay. So how many of you, and this will be, I think, maybe an illustrative thing. So how many of you feel like that you got yesterday, that you got either about 10 minutes of vigorous intensity physical activity, or you got about 20 to 22 minutes of moderate intensity physical activity, or some kind of combination of both of those things, like sort of wave at me or flash your camera or something. Okay. All right. So almost all of you guys did that. Okay. And that's pretty standard. Okay. Pretty standard. You guys, if yesterday was a reasonably indicative day, a reasonably representative day for you, then you all probably get your 150 minutes of moderate or 75 minutes of vigorous, or some kind of combination of those two things over the course of a week. Okay. You guys are going to get enough activity that you're probably going to gain some health benefits from all of this. Okay. What I'm going to tell you now, though, may frighten you. It should frighten you. Okay. As a group, college-aged individuals, you guys are as active right now as you're ever going to be for the entirety of your life. Okay. Think about how active your parents are. Think about how active your grandparents are, and things like that. Okay. You guys are as active now as you're ever going to be. And I mean, that there may be some exceptions from people that are relatively sedentary as college age students, but your environment is set up. Your world on campus tends to be set up to where it's easier for you to walk around and get activity, or go play intramurals, or, do things with your friends. Your environment is set up to do all of that. Normal suburban American life and job situation is not set up for that to be the case. Okay. Especially for people that are like you guys. Okay. You're very likely going to have college educations. The vast majority of people that have college degrees work in jobs where you sit around all day. And that may not always be true. Some of you guys want to go into allied health. You're going to be a doctor or a nurse or a physical therapist or something like that. You may be up on your feet doing some things, but you're getting light intensity activity, which is good, but you're not doing a lot of kind of moderate or vigorous intensity activity as part of your job. Okay. So when we look at this increase in these chronic diseases that are associated with less physical activity, when we look at the increase in obesity rates that have exploded in the last, say, 25 or 30 years, I want all of you to keep in mind that some of this is just a structural problem. Okay. If you think about 1900, let's just say, okay, 1900. What do you guys think was the most common job in the US in 1900? Anybody want to guess? Jordan, what do you think was the most popular job, or the most common job? Popular maybe is the wrong connotation. The most common job in the US in 1900 was what? I'm sorry. You got real garbly on me. Can you try that again, please?
Mining or like digging or something like that.
I apologize. I still didn't catch you. Are the rest of you guys, is the problem on my end, or is it on y'all's end? It's my end.
I heard him.
You heard him? Okay. What did he say, Savannah? I'm sorry.
He said mine rigs.
Okay. Yeah, mine rigs, farming, right? Anybody? My grandfather, my mom grew up on a dairy farm in Kansas, and let me just tell you guys, like, farming involves a lot of strenuous activity and work and those kinds of things, right? You were farmers, or you worked in a factory, or you were doing mining or something, like stuff that was manual labor, right? Things that we do now, not that way. Okay. When we go and we look at developing nations, their most common kinds of employment involves lots of physical labor. And so these are structural problems that are only going to get more and more pronounced as we automate everything. Okay. So just keep all of that in mind. But it's not that hard. It takes 10 minutes a day. Go for a run. Go run for 10 minutes, right? If you have a dog, your dog would love to go for a run for 10 minutes. It's not, you don't have to run that fast, okay? If you don't have a dog, come to Oklahoma City and you can run my dogs for like 10 minutes at a time. They would adore you if you did something like that, okay? All right, so I hope that from some of this, and we're going to do a couple of little things at the very end here, and then we're going to kind of wrap up some of this idea of physical activity and obesity and health. I want to talk very, very briefly about a couple of more things that are going to crop back up for us when we talk about prescribing exercise and things that are going to be important. It's going to get at this idea of how are we trying to quantify the amount of activity and how are we trying to quantify the intensity of activity. Okay, so what I'm going to do is I'm going to share my screen. And so we're going to go here and then here. Okay, so I'm going to try to write on the screen again, guys. Everybody can see my screen. Y'all can see the PowerPoint and everything. It's up. Okay, cool. So if we remember from yesterday, we talked about, there we go, come on, that we oftentimes break intensities into light, moderate, and vigorous, okay? And so that is all going to be based, and these intensities are all going to be based upon the relative change in energy expenditure that we have once you start moving around and doing new things, okay? So you may remember from yesterday that I talked about that we have those big fancy machines, the metabolic carts in the lab, where we can directly measure how much oxygen you're using, and we can calculate something called VO2 max, okay? So the single best way for us to define, are you doing light, moderate, or vigorous intensity physical activity, is to measure your energy expenditure or measure the change in energy expenditure and express it relative to, like, as a percentage of sort of your highest possible energy expenditure during exercise, which would be this VO2 max measurement, okay? So we would have Gabby, we're going to have her go and run, and we're going to measure, and we're going to say, okay, she is expending energy that is roughly half of what her maximal thing is, okay? And there are some kind of very rough things that are laid out, you know, that like light intensity activity is maybe 0 to about 35% of VO2 max, right? The change is going to be about there. Moderate is going to be about 35 to maybe 60 or 65. And then vigorous is going to tend to be 60 or 65 to about 100% of VO2 max, okay? As we talked about yesterday, though, we don't have these pieces of equipment readily available. They're very expensive. They're very cumbersome. I can't chase Gabby around with my computer on a wheelie cart, like as she runs around and things. So it's not a very practical way for us to do it. Okay. Even if it's the best way for us to say, hey, you're going to go exercise today. I need you, if you want to be doing moderate intensity physical activity, I need you to be at this amount of energy expenditure. Okay. So what we tend to do, rather than doing that, to get some more practical information, that if you guys have an Apple Watch, or if you have a Fitbit watch, or something like that, or if you own a heart rate monitor, which you can buy them relatively inexpensively, is we can do things where we try to give you, and we look at how high is your heart rate going during a particular bout of exercise, okay? And we're not going to get into kind of the intricate details of the differences between looking at whether or not we're expressing your heart rate as a percentage of your age predicted max, or something called heart rate reserve. I'll talk much, much more about this when we get into prescribing aerobic exercise, but I just want you guys to appreciate that I can track your heart rate during whatever it is that you're doing. And there are some kind of relatively specific laid out percentages of heart rate, either as a percentage of your max heart rate. So it's gone up this much, right? You need to be at 120 beats per minute, or you need to be at about 140 beats per minute or something like all of that to kind of track whatever it is that we're doing. Okay. And so that's one of the reasons that we can use heart rate. That's one of the things that's going to go on, as we talked about yesterday, that these kind of fitness trackers are going to be able to do, because the percentage of your heart rate, the amount that heart rate changes, is very, very strongly related to the amount of change in energy expenditure that we're going to see. Okay. So for any of you that did something yesterday, if you were wearing, say, an Apple Watch or a Fitbit while you were doing things, then it should tell you like what your heart rate change was going to be while you were doing things. And then it can say, okay, it thinks you were walking and your average heart rate during that 12 minutes of walking was something. And so then we can use that to try to approximate, was it light intensity walking? Was it moderate intensity walking? Okay, so those are some things that we can do. So the last thing that I want to bring up is a slightly less intuitive way of trying to estimate exercise intensity, but it's also a very useful kind of thing that we can do, and it's something called a metabolic equivalent, or a MET, an MET. Okay, so if physical activity is anything that raises energy expenditure above resting energy expenditure, then if we know your resting energy expenditure, we could couch anything that you're doing as being some amount relatively higher than rest. Okay. And so a metabolic equivalent, they tend to be expressed in sort of whole numbers relative to resting rate. So a MET value of one metabolic equivalent, is equal to whatever you guys are doing with your resting metabolic rate. Okay, so any of you, I'm trying to remember who it was that had flashed their camera earlier that seemed to be like laying down on their couch, right? You're at one MET right now. If I have you get up and start doing jumping jacks and it raises your energy expenditure to be, say, 3 times your resting rate, then you would be at 3 metabolic equivalents. Okay. Or you go and you're running and you're at 9 times your resting metabolic rate, then you're at 9 METs or something like that. Okay. And so everything with a MET is expressed relative. So 1 MET is resting metabolic rate. It's all expressed relative to that. Okay. So one of the original ways that people laid out light, moderate, and vigorous intensity physical activity was by looking at these MET values. And so light activities are MET values that are less than 3 times your resting metabolic rate. Okay. And that tends to be walking slowly around home or the office or something like that. Moderate intensity physical activity is maybe 3 to 6 METs. Okay. So you're doing something where your metabolic rate or your energy expenditure is 3 to 6 times resting rate. And then vigorous is anything that's over, say, 6 times your resting metabolic rate. Okay. We'll bring some of this back up a little bit more. So metabolic equivalents were much more popular and much more widely used in, say, the 90s and the early 2000s, kind of when I was in graduate school in the early 2000s. They have in some ways fallen a little bit more out of favor now because of the advent of our ability to use these fitness trackers much more so. The technology for monitoring heart rate and these kinds of things has become a lot less expensive. And so now we're much more able to kind of get a heart rate value, which makes this kind of use of these MET equivalents a little bit obsolete. But I do want to bring it up because there are some very popular questionnaires that we can give to people that you guys may use for your capstone project or something, where it asks people kind of what are you doing, and then it uses these MET values to try to define like, okay, gardening has a MET value of 2.5, or jogging tends to have a MET value of 8 or something, and you can calculate some energy expenditure from those things. So I just like to introduce it. Okay. Okay. So the last little bit of things that I want to emphasize for you guys, okay, is that while you guys, almost every one of you, got a sufficient amount of exercise or physical activity yesterday, that if you did that every day, you would meet the physical activity guidelines, okay? But you guys are not the norm. So imagine if you all get counted in the sort of US adult population, okay, that about 70% of adults do not get enough physical activity to meet the recommendations. That includes college students, okay? So imagine if you all are easily the most active. Imagine what it looks like for people that are in my age bracket, right? I'm 43, so imagine people that are like me or your parents or something. We're not at 70% of us that don't meet the guidelines. We're at something more like 80 or 85%, okay, that don't meet the physical activity guidelines. And then the really staggering group to me is that there are somewhere between maybe 30 and 40% of adults that have no leisure time physical activity whatsoever, okay? Outside of anything that they do from a job standpoint or whatever, they get no leisure time physical activity. They do nothing. They get up in the morning, they go to work, they come home, they sit on their butt, they watch TV, they do nothing else, okay? And so that's a problem. It's really, really, really a problem. And this is one of the things right here that is absolutely contributing to the prevalence of all of our chronic diseases and those kinds of things. Okay. We need to be better about this. We need to find some ways to try to combat some of this. Okay. So I do want you guys, right, to kind of know a little bit about some of these numbers and this kind of thing. Okay. Okay. Here are county level estimates of physical inactivity. The darker blue is the larger the percentage of people that are not meeting, that are not getting any sort of leisure time physical activity. And so you can note on here, right, everything looks in sort of this area in the southeast and up into what we would kind of refer to as Appalachia here through Kentucky and West Virginia, right? And these kinds of things. You'll note how much more active people tend to be in the Northeast and on the West Coast, and those types of things, all right? If you look at this and we were to overlay the prevalence of cardiovascular disease, the prevalence of obesity and things, they tend to mirror each other really, really well in the midst of all of this. And so we have lots of data. We know where the problems are. We know what's going on, but we don't have very good solutions for all of this. Okay, so still these kinds of things. This is the kind of last little bit of this that I want to do, and then we'll move on and we'll actually talk about some physiology today and kind of start getting into kind of how we're going to determine some fitness and what normal function is, and then how inactivity is going to mess those kinds of things up. Inactivity, not meeting the physical activity guidelines, is considered to be a risk factor, okay? It's a behavioral risk factor for the development of chronic diseases, all right? And so I want you all to look at these kinds of things. If we look at this, okay, this is leading risk factors for global mortality. This is from 2004. These data are pretty old in that way. But if we look at this, tobacco use, okay, and physical inactivity are considered to be what we would call they're not. These are considered to be, I apologize, my stylus is not working very well. Tobacco use and physical activity are considered to be modifiable risk factors. These are things we can do something about, okay, in theory, Okay, we have some level of choice. Some people just have high blood pressure. Some high blood pressure can be modifiable, some isn't. High blood glucose, these things tend to be here. So if we look at this, physical inactivity is the second leading modifiable risk factor for all-cause mortality around the world, okay? You guys probably have heard your entire life, right? Don't smoke, don't smoke, don't use tobacco, don't use tobacco, do all of these things, right? There's all of this funding from TSET in places where the tobacco companies were, we'll just say, disingenuous at how dangerous cigarettes were, how addictive nicotine was, and those kinds of things. How many of you have heard all kinds of commercials on TV and other big pushes about, hey, like maybe don't be physically inactive? Anybody? Right? We don't make a big deal about all of this, but this is the second leading modifiable cause. And as tobacco use falls, then at some point we are likely to get to a place where being physically inactive is the leading modifiable risk factor for all-cause mortality all over the world. Okay, so this is a big deal, and we don't make a big enough deal about it kind of when we're in elementary school and in junior high and those kinds of places. Okay, so interesting things that maybe you guys want to care about. Being physically inactive costs about $150 billion in healthcare cost every year. If any of you guys have tried to get your own insurance, or maybe if you're on your parents' insurance and you have any kind of idea about how expensive insurance can be, one of the reasons it's expensive is that 10% of the people, about 10% of the population accounts for 80 to 90% of all healthcare costs. And so you guys are not the people that are using healthcare cost. It's the people that are inactive. It's the people that are obese. It's the people that are older and have these chronic diseases. So this is a big number. If we could modify some of that, everybody's insurance costs less, and that can be a good thing. Per person, people that are physically inactive tend to spend about $1,500 more per year going to the doctor than people that are active enough. It's about 42% more in that particular way. I don't know about you guys, but I can think of a lot of things I would rather do with $1,500 than give it to a doctor, right? If all I had to do was go out and go for a walk every day, and I could potentially reduce some of those kinds of things, right? So you can have a pretty nice time at the beach for $1,500 or something like that. Lots of other things that we can do there. Okay. So many of you may have been thinking, Dr. Black, this is great. We know all of this, but why in the hell is being inactive so bad for us? Like, what's the problem with all of this? And the answer is that, unfortunately, we have some ideas, but we really don't know. Okay. We really don't know. We think there has been a confluence of evolution, genetics, along with kind of changes in modern society that are all working together. One of the best kind of working hypotheses that we have is that we evolved, our genetics, we're really, really good as humans at storing energy. So you eat food, and we're really, really good at storing a lot of that extra food that we have. And we think some of this has come out of the time when, as a society, we were hunter-gatherers. And so there was a lot of instances where you had to go hunt today or gather today in order to be able to eat today. And so there might be these kind of alternating cycles of, we found food or we killed food today, so we get to eat today and tomorrow, but then there might be a couple of days when we might not be able to eat. But we needed to be able to store food from when we could eat so that we would have energy to continue to try to hunt and gather. And so if you look at things kind of like with us and other animals, we're really, really efficient at, when you eat excess food, we're really good at storing it as adipose tissue and fat and holding on to it for the next kind of period of time. There's some positives to that, but for a lot of people now, right, we have removed a lot of this kind of selective pressure from those kinds of things. For maybe 80, 75 or 80% of the population, we tend to have pretty reasonable access to food every day. Okay. Not that poverty and food insecurity is not a huge, huge issue, but for a majority of people in the US, we get enough to eat every day. In a lot of instances, we probably eat too much, right? We can go to Chick-fil-A, we can go to CC's Pizza and do all you can eat. We have fried foods, we have things that are going to be very, very calorically dense. That's a little bit different than kind of how things were when our genes evolved. And so when you combine excess food and very nutrient dense food, or sort of calorically dense food, with no need to go walk around or run around to chase an animal down and hunt, or to go walk around to try to be able to gather and find fruits and vegetables and those kinds of things. We sit behind a desk all day. We're much, much less active, and we have easy access to calorically dense food. And that is a very, very deadly combination in some ways. And so there are people that will argue that the human genes, that we are the normal function to get good function of our genes, we need to be physically active, and we are no longer physically active in that way. And that's why getting out, walking, exercising, the people that are more active, they should be the quote unquote normal ones, and the ones that are sedentary, that's actually the thing that's causing a lot of problems. And so this evolves. We get new data every year on some of this, but this is kind of the best working hypothesis that we have. Is that our kind of lifestyle these days does not match up with the way our genes kind of store food and metabolize things. And we'll talk more about kind of the ins and outs of that as we move along. Okay, so we are going to do this. I'm going to stop sharing. So we are recording again. I'm going to share my screen. All right, so we're going to break this up into a couple of different lectures, and kind of contained within this one kind of big long PowerPoint. There's really going to be some anatomy and physiology of muscles, some anatomy and physiology of the neuromuscular system, so kind of how the brain communicates with muscles, as well as some of the physiology on how do we contract our muscles, how do we generate force, how does all of that work, and then a little bit at the very end about how muscles might respond to increases in activity. So there's a lot of stuff in here, okay? We're not going to cover all of that today, and probably even tomorrow. It will probably bleed into a little bit on Friday, okay? Okay, so some very simple kind of starting off learning objectives for all of this. I want you to know the basic structure and organization of skeletal muscle. I'm really, really hopeful that much of what this is things that you guys have seen at least once, if not multiple times, before my class. So I'm just going to kind of hit some of the high points on all of this. There's more information in the PowerPoints. I do want you to know most of the stuff that's in the PowerPoint that's also mentioned on the study guide, but I'm just going to kind of hit some of the high points. I'm not going to go detail by detail by detail into all of this, because hopefully you guys have seen much of this before. We're going to talk about the sliding filament theory of contraction. So how do muscles contract? Because when muscles contract, we're going to use energy, we're going to generate force, we're going to generate movement. And so that's going to be one of the key things that plays a role in our ability to have physical activity. And that increase in energy expenditure when muscles are contracting is going to be one of the things that drives some of the adaptations, both either from resistance training or from doing endurance training, okay? We'll talk some about motor neurons and motor nerves. So how does the brain tell your muscles to contract? And all of those things, even though I can't see people on their cameras, but if I was to say something like, Erin, wave at us, and then she waves at us and she does all of this, there's a whole sort of signaling process that's going on where her brain thinks about waving, and then she thinks about it and she sends a bunch of signals, and then her muscles contract in her shoulder and her hand and things, and then she does the wave. So we'll talk about some of that and how all of that goes. I also really want you guys to appreciate how structure and function are going to be linked. And this is going to be really, really important for those of you that want to go into, say, personal training or strength and conditioning as a thing, or physical therapy, and even medicine to some extent. But the way in which we move is very much dictated by the shape and location of various muscles. It's very much dictated by sort of tendons and ligaments and joint structures and those kinds of things. And so we need to understand and appreciate how all of that works together to let us generate movement. And so that when we have impairments and things, when people are either sedentary or they've had an injury, or if you have weaknesses in certain muscles that are then gonna cause changes in that function, that we can kind of understand those things and then use our knowledge of structure and normal function to be able to intervene in some ways and try to fix things and make people healthier. Okay, so really quickly, these are the three basic types of muscle. The one that we're going to spend all of our time talking about today is going to be what we call skeletal muscle. Okay. Skeletal muscle is unique from cardiac muscle and from smooth muscle in a couple of ways. The big thing about skeletal muscle is that it's what we call striated. And so when you look at skeletal muscle under a microscope, it has striations or stripes, okay? And these stripes are due to the distribution of filaments made up of actin and myosin. And so we'll see some pictures of all of that in the coming slides. But it's very much, it looks a certain way because of the way that the structures line up. And so these striations allow us to pack a bunch of actin and myosin in together. And the more actin and myosin we have, the more contraction force we can generate. And so it's really important in this way. The way that they're organized is going to help us impart some function. Okay. Cardiac muscle is also striated, but it's going to be a little bit different than skeletal muscle. Smooth muscle is not striated. Smooth muscle is a big old hot mess. It's sort of actin and myosin filaments are just running all over the place, but it performs a function in your digestive system and around your blood vessels in some different ways. And so it doesn't need the striations. The other really important thing that makes skeletal muscle different from cardiac muscle and from smooth muscle is that it is what we call voluntary. Okay. And by voluntary, what I mean is you can consciously control the muscle. You can make it contract within reason when you want it to. You can make it stop contracting when you want it to. Now, there are some instances when that's not always going to be the case, but it's very voluntary. Okay, so your cardiac muscle, you guys, and this is a good thing, right? You don't have to think about and tell your heart to contract every time that it beats. Okay, you don't have to. With smooth muscle, you don't have to think about making the smooth muscle that surrounds your small intestine and your large intestine and things contract to help move food through the system in those particular ways. Okay, so that's one of the things that makes skeletal muscle very, very unique. Okay, striations, and it's voluntary. And you're going to have some special structures that are going to help us with do all of those kinds of things. Okay, I have a weird question for everybody. So, does anybody know what a Russian nesting doll is? Okay, Jessica, you're nodding your head. What is a Russian nesting doll?
Like a Russian nesting doll is like where it has like a big outer shell. You take it off, there's another doll. You take that one apart, another doll inside, and it just keeps going till there's a tiny little one on the inside.
Okay. What do they look like? What does each one of them look like?
Like they normally look similar to each other, but just a little different.
No, like, perfect. That's all that else. They're meant to be in many ways, right? Like almost identical. Okay. So those dolls, you break them open and there's like a smaller one inside, and then you break that one open and another small one until you get down to the very, very kind of smallest one. Okay. That's what a Russian nesting doll is. The way that I conceptualize how skeletal muscle is organized from its smallest to largest structure is that if you think of skeletal muscle as a Russian nesting doll of tubes, basically, okay, of tubes or filaments or something, that's what skeletal muscle is going to be like, okay? It depending upon on which end that you start. If let's say we start on the largest end of things, okay? Oops, I'm gonna go back, clicked on the wrong thing. If you start at a whole muscle, okay? And so I'm gonna apologize for this. Don't laugh, but if we take my bicep, okay? We take my bicep, all right? It's right here, right? That's a whole muscle, all right? Whole muscle. If you cut open my bicep, you would see that there are all of these kind of nearly identical, smaller tube-like structures or bag-like structures that are going to be contained inside my big, kind of bigger tube muscle, okay? Those are called fascicles, okay? And so if you cut open a fascicle, you would see that the fascicles have a bunch of smaller tubes in them that are called muscle fibers, okay? So here you have a whole muscle. Here you have each one of these things is a fascicle. We pull out one fascicle, we cut it open. You will see that there are then all of these individual muscle fibers that are going to be in there. A muscle fiber is a cell, okay? One fiber is one muscle cell, okay? Muscle fibers are some of, if not the largest individual cells in your entire body, okay? I could cut out and show you one single muscle fiber and you could see it without needing a microscope, okay? It might be somewhere from a couple to maybe 5 or 6 or 8 centimeters long, okay, depending upon what muscle we took it from. So you can see this individual cell with the naked eye. So they're these giant, giant cells. And there's going to be some individual kind of properties of them that we're going to have to talk about because they're so big, okay? So whole muscle, fascicle, made up of many fascicles. Each fascicle is made up of many, many muscle fibers. If you cut open one muscle fiber, you would see that inside each fiber is a collection of smaller tubes that are called myofibrils. If you then cut open a myofibril, you would see that a myofibril is made up of these individual little structures that are called sarcomeres, and it's a sarcomere and then another sarcomere and another sarcomere. They're kind of stacked end to end. And then a sarcomere is made up of these filaments of what we call contractile proteins of actin and myosin. And I'll show you guys some pictures of those. But you're basically going from these strings of protein that are then going to be kind of organized into a sort of smaller little kind of baby little tube thing. And then we stack all of those end to end and they make a bigger tube. And then each of those bigger tubes, those myofibrils, we're going to have multiple of those inside one fiber, which is also a big tube. And there's many fibers in a fascicle and then many fascicles that are going to make up a whole muscle, okay? So that's this idea of what I like to think of as kind of this Russian nesting doll of tubes that make up skeletal muscle. Each individual sort of area here, okay? A whole muscle, a fascicle, the individual fibers, especially, maybe not so much the myofibrils and the sarcomeres, but these first three, each tube is going to be surrounded by connective tissue. And you don't have to know this, but it's called the epimysium, the paramysium, and the endomysium. But it's connective tissue. So it's got collagen and stuff around it, okay? So it's stretchy. And then when you get down at the sarcomere level and at the contractile protein level, there's going to be some other connective tissue. They're going to help kind of hold all of those things together. And so the good thing about having all of this connective tissue is going to, at some point on the whole muscle level, come together and make a tendon. Okay. And then that tendon is going to attach the muscle to the bone. And so what happens because of all of this connective tissue is that when things happen at the protein level, when we get movement, when myosin interacts with actin, and I'll show you guys some of this in a few slides, when myosin interacts with actin and it pulls on these actin filaments, as the actin filament moves and slides, that pulls on some connective tissue at the sarcomere level, which pulls on connective tissue at the myofibril level, at the fiber level, at the fascicle level, all the way up to the whole muscle level. And so, which then pulls on the tendon and creates movement. And so when you do a bicep curl or you do any kind of muscle contraction, what's going on is there are these changes in things that are happening all the way down at the level of the proteins in the muscles. And the proteins are what interact and move, and they keep pulling on things, which eventually gets transmitted out down the tendon and leads to movement. Okay. So what you see happening at the whole muscle level is the consequence of interactions at the microscopic level between individual proteins of actin and myosin, okay? And so when there are problems, either with the connective tissue or with things that are going on at the protein level and how they're going to interact, then that can lead to problems with being able to generate force, with being able to contract your muscles, et cetera, et cetera. Okay, so we need to kind of appreciate how all of this is organized, because the one thing that we're trying to do here, the single thing, is we want to create a situation whereby we can pack as much contractile protein into as small an area as we possibly can. And we need to keep all of it very tightly organized so that all of the actin and myosin are going to interact and pull together in the same direction so that the whole muscle can function properly, okay? And that's one of the reasons why muscles are organized the way that they are. We need as much contractile protein as we can get, and we need all of it to pull in the same direction. Because if I had bicep stuff where some of it's pulling this way and some's pulling this other way, then they cancel each other out and no movement ever happens. Okay? Does that make a level of sense for you guys? Okay. All right, I can see Jessica nodding her head. That's good. At least one of you guys has I've not confused yet. So, okay. A couple of other things now. We're gonna get on a couple of other structures that are going to be really important for how muscles contract. Okay. So what you can see here, this is a cross section, or kind of a cartoon of a cross section of an individual muscle fiber. So this is one muscle cell. Okay. And so what you can see here, right, this is a myofibril, this is a myofibril. You can see all of those things that are going to be in there, okay? If you look on the myofibril, it's going to run from about right here to this place right about here. This thing, this area here to here, this is a sarcomere, okay? So, and then these lines in the sarcomere are going to be contractile proteins. But at the fiber level, what I want you guys to appreciate is that the surface of the muscle fiber out here, the cell membrane, is called the sarcolemma. Sarco just means muscle. I forget if it's Latin or if it's Greek, but it means muscle, okay? So it's the membrane of the muscle, all right? On the membrane of the muscle, there are going to be little holes, and those holes lead into tunnels that are going to go from the outside and be connected here on the outside of the muscle membrane of the sarcolemma, and they're going to go all the way down here into the inside, where they're going to come into very close contact with the sarcomeres and the myofibrils. This tunnel is called a transverse, or a T-tubule, okay? And we have to have these T-tubules in order to move the signal from the nervous system, from the outside of the muscle fiber down into the inside, so that we can release something. We're going to release calcium from the sarcoplasmic reticulum, which is this kind of dark, kind of reddy maroon structure here, okay? It's just basically a storage closet that is going to have this network of things that's going to have a bunch of calcium in it, that's going to web its way around all of the myofibrils and sarcomeres. And so the T-tubules are going to be connected here to the sarcoplasmic reticulum. And what's going to happen is your nervous system is going to transmit a signal. That signal is going to get onto the membrane, onto the sarcolemma. It's going to then move along the sarcolemma and come down the T-tubule. And then the T-tubule is going to tell the sarcoplasmic reticulum to release calcium. And that calcium is going to go out and it's going to interact with these contractile proteins. It's going to interact with troponin, and it's going to cause actin and myosin to do things on the inside of a muscle cell. Okay. And then when we want to turn it off, the calcium goes back into the sarcoplasmic reticulum, and we shut off our signal, and it turns things off. Okay. So you guys need to know what those things are. At the cell level, at the fiber level, we're also going to have mitochondria here, 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 okay? Mitochondria are the place where we make ATP. And so they're going to use oxygen, and then they're going to also use glucose or free fatty acids or something. They're going to make ATP, and we're going to need that ATP to allow actin and myosin to contract. Additionally, and this is one thing that makes muscle cells unique, muscle cells have not one, but many, many, many nuclei. Okay, because the cells are so big, they need multiple nuclei to be able to supply all of the proteins and enzymes and things that the cell needs to function correctly. So skeletal muscle is what we call multinucleated. Other things that are unique about the nucleus is its location. In skeletal muscle, the nuclei are actually going to be located out here at the edge. They're not located in the middle like we tend to see in most other cells in the body. And this is partially due to the fact that they want to maximize as much space in the middle as they can for actin and myosin and the contractile machinery. So when you look at them under the microscope, they look a little weird and a little strange. And you're kind of like, why are the nuclei out here on the outside when you would expect them to be on the inside? Okay. Okay. So this is as good a place as any. Let's take a 5-minute break. We'll come back at 11:50. Okay. I'm going to pause the recording, resume recording, back to the muscles. Okay. Back to the muscles. For it to be a podcast, I need like some cool like mood music in the background, right? And you're like, what if I told you that muscles were the coolest thing in the world? Now let's hear from so-and-so. And you get a little bit of music, and we get a little story. I need some production value. Okay. So I think I covered most of those key points. So in the myofibril, so we've taken our muscle fiber, we've pulled out one myofibril here. In that myofibril, you can see 1, 2, and then 3 sarcomeres that are going to make up that individual myofibril. Okay. So inside the sarcomere, you see these little lines that are going to be denoted like this, and then you see this line right here. These lines are thick and thin filaments. The thin filaments are made up of myosin. The thick filaments are made up of, I'm sorry, I got that backwards. The thin filaments are made up of actin, and the thick filaments are made up of myosin. And then the place where one sarcomere connects to the adjacent sarcomere here is called the Z line or the Z disc. Okay. And there's some other structural proteins that help anchor everything down that you guys will learn about in exercise phys that are going to go on there. Okay. But it's at this place where actin and myosin are going to overlap with each other and are going to be able to make contact and interact. That's where all movement comes from. That's where it's the interactions of those proteins. So all movement, all strength, everything comes from what's happening inside the sarcomeres with the interactions of actin and myosin. Okay. So we've done most of these kinds of things. Here is a better view and a better blow up of what's going on at the level of the sarcomere. Okay. So we have a Z line here, and then a Z line over here on the other side. And then in between those, you can see what the filaments are going to look like. Okay. So thick filaments that are made up of myosin. So this filament that's right here, it's called thick because the myosin is a bigger protein than actin. It has a larger molecular weight. And so when you look at it under the microscope, it looks darker, right? It looks bigger and fuzzier. An individual myosin protein, let me see if I can move this around. The individual myosin protein looks like this, okay? And I absolutely hate the way that this kind of thing looks. It's not maybe the most appealing thing, but it's basically, it has a hinge on it. And so you've got this tail portion of the myosin, and then it has kind of a forked head region that comes off of it. And so in the thick filament, all of the tails of the myosin are going to be linked together. And then at these very precise intervals, coming off of the thick filament are going to be these head regions that are going to be like this. And so it's the head region that can make contact with and connect onto the actin filament. And then you've got your myosin molecule, right? And it looks like this. And then it's going to grab on to actin, and it's going to fold back on itself. It's going to pull backwards. And as the head tilts and moves, then it's going to pull that filament along with it. Okay. So at the sarcomere level, all of the myosin here on the right side, so all the myosin on this side of the sarcomere, when their heads want to tilt, they're going to want to pull everything in towards the middle this way. And then all of the ones over here on what you guys can see as the left side are going to tilt and try to pull the myosin or the actin filament in this direction. So if you think of a sarcomere as looking like this, when it contracts, it's going to pull in on itself and pull these Z disc or these Z lines towards each other. Okay. As one sarcomere does this, it pulls on its Z disc, and that's going to try to pull the adjacent sarcomeres along with it. And if you can get multiple sarcomeres, right, you've got millions and millions of them in a muscle. If you get many, many of them all doing the same thing at the same time, all of these things in your muscle are going to pull in on themselves. That's going to shorten the muscle. So as I move my bicep like this, all of my sarcomeres are squeezing in together, okay, or many of them are squeezing in together. They all pull on the adjacent one, which eventually pulls on the tendon, and that's going to give us this shortening movement that we call a concentric muscle contraction, okay? I'm sure that you guys have seen and kind of gone into great depth about all of this in anatomy and physiology, correct? You guys have seen all of this before? Okay. I really just want to hit some of the high points. If you really, really want to know all the nitty gritty about this, you can get in our master's program and you could take advanced exercise phys with me or neuromuscular physiology with me. And we'll go into all of the individual subunits of the actin molecules and the myosin molecules and why Emma has different genetic things that are going to make her actin and myosin slightly different than Gabby's, which are slightly different than Savannah's, and how all of that imparts different levels of strength and speed and function. But that's kind of beyond what I want us to do here today. Okay. So the other piece of this, okay, we've talked about some of the individual structures. We've talked about how they're going to move together. But I also want you guys to understand a couple of other things about muscle fibers and some of the structures that are in those fibers, okay? Not all fibers are the same, okay? They mostly look the same and they mostly behave in the same way in how they contract, but there are some properties of the myosin molecules and the mitochondria and the sarcoplasmic reticulum within individual muscle fibers that are going to be a little bit different. And so the distribution of these different kinds of fibers within a given person is going to play some role in determining, are they stronger than someone else? Are they faster? Can they jump higher? Can they recover faster between contractions and those kinds of things, okay? So one of the things that is going to be different between individual muscle fibers is going to be what we call the biochemical properties of them. Anybody had biochemistry? Emma has. You survived biochemistry? I still have nightmares of biochemistry from like 25 years ago in that particular way. Do they make you hear? Do you have to like memorize the molecular structure of all of the amino acids and all of how like all of the structures of all the things in like glycolysis and the Krebs cycle and all that bullshit?
Depends on who you have. Structures of amino acids, yes. Structures of everything in glycolysis and Krebs, maybe. Okay. But I like biochem. I'm a little crazy.
Well, I like the idea of biochemistry. Like I like a good signaling pathway. Like I like the steps and things, but I sometimes would get confused on that we've got an OH chain here versus, you got a hydroxy here versus something else over here. And this is methylated. And I want to scream. Anyway, there are differences in the biochemical properties of certain muscle fibers. Okay. One of the things that is going to be different, and I guess I should back up, but I don't know why this is not in the right order. I'm going to go forward a slide and then I'll come back to this other thing. I probably should start with this. Maybe this thing just got moved out of order for some reason that I wasn't appreciating. Muscle fibers come in 3 distinct types, okay? 3 distinct types. And those types are determined based upon something called their MHC isoform. MHC stands for myosin heavy chain. Okay. We're not going to talk about where the myosin heavy chain is. We're not going to talk about what the myosin heavy chain is. We're not going to talk about what the myosin heavy chain does. All I want you to know is that there is a piece of the myosin protein that can be different among these different muscle fiber types, okay? And so based upon this isoform, a muscle fiber can either be a type 1 muscle, a fiber, a type 2A muscle fiber, or what we call a type 2X fiber, okay? Type 1 muscle fibers are sometimes called slow twitch muscle fibers. And then type 2A and type 2X are sometimes called fast twitch muscle fibers. And there's a lot of history as to why they are called fast and slow, or why they are named based upon their MHC isoform, but it's really beyond the scope of this particular class, okay? So in your muscle, okay, in your muscle, if we were to look in our muscles, all right, I'm trying to pick out somebody here. So like I see John on my screen. If we were to look in John's muscle, okay, John, like, let me see your forearm or something. Let me do this here. We're gonna talk about this. Okay, we look in John's forearm. We pull out one of his forearm flexors. We dissect it down. In that muscle, all of his muscle fibers, right, are going to be one of these three types. That's not 100% correct, but it's correct enough for our purposes in class. He's going to have some distribution. Some percentage will be type 1, some percentage will be type 2, some percentage will be type 2X, okay? Thank you, John. John's percentage is determined by genetics, okay? And so then if we were to look at, say, on my screen, Nia is next to him. We were to look in Nia's muscle. Her percentages are also determined by genetics, and they're likely going to be different than what John's are, okay? And the distribution of these things in John's forearm are going to be relatively similar to the distribution in his bicep and his quads and his calves, okay? So everyone has some genetically predetermined fiber type distribution within their muscles. Now, your calf muscles and your diaphragm and things are going to tend to have more type 1 or slow twitch fibers than, say, your glutes or your quads or the muscles that go to your eyelids and those kinds of things. But in general, if you have a lot of type 1 or a lot of slow twitch fibers in one muscle, you're going to have a lot in your other muscles. If you have a lot of type 2 muscle fibers in one muscle, you're going to have a lot in the other ones, okay? So this is mostly genetically determined. We can't do anything about this. So things to keep in mind, okay? Type 1 muscle fibers have, I'm going to write this, oh crap. Type 1 fibers, okay, tend to have very high levels of capillaries, very high levels of mitochondria, okay, in comparison to type 2. So if we look at type 2, they have lower capillaries. They have lower amounts of mitochondria, okay? Whereas type 2 fibers tend to have higher levels of sarcoplasmic reticulum, okay? And type 1s may have lower levels of, say, sarcoplasmic reticulum. And so the consequence of all of this is really, really important, okay? It's going to be that we say that type 1 muscle fibers have a higher, what we would call oxidative capacity. Type 1 muscle fibers, because they have more capillaries, they can deliver more oxygen to those muscles. And because they have more mitochondria, they can make more ATP using that oxygen than their corresponding type 2A or type 2X muscle fiber types can. Because of that, type 1 muscle fibers can generally tend to have better endurance than type 2 muscle fibers do, okay? So if you think about this from an athletic standpoint, people that tend to be better endurance athletes, better distance runners or cyclists or something, especially on the elite, say collegiate level or Olympic level, they're going to have much, much higher levels of type 1 muscle fibers genetically than people that are, say, very, very good sprinters, okay? If you have lots of type 2 fibers, be they 2A or 2X, you're going to be able to generate force faster. You're going to be able to generate more force in some ways because you can release more calcium because you have more SR. And so people that are type 2 kind of predominant tend to run very fast, jump very high. They're these kind of explosive athletes, okay? Now, most of us, okay, and I know that we have several of you guys that are collegiate athletes in class. Good for you guys. Y'all are, by definition of being at this level, very likely to be kind of genetic outliers from a kind of fiber type or a functional standpoint. The rest of us normal people, okay, we tend to be, most normal people are going to be somewhere between 40 to 60% type 1 versus type 2 in one direction or the other. The vast majority of people are pretty equal and kind of in their fiber type distribution, okay? And that's why we're all kind of average in our ability to kind of how far we can run and how fast we can run and those kinds of things, okay? Nia, I see your question. Let me do one more thing really quickly, and then I'm going to come to you, okay? The other thing that is different, okay, the other thing that is different is that there is an enzyme that's contained on the myosin heavy chain, okay? And that enzyme is called myosin ATPase, okay? And so if you have a type 1 myosin heavy chain, your myosin ATPase, it's an enzyme that splits ATP down. It breaks it in half. It helps us use energy really, really quickly. Type 1 fibers have a slower myosin ATPase, so it breaks ATP down slower than the type 2 myosin ATPase does, okay? And so because of all of that, that's where we're going to get speed of contraction, okay? We get endurance capacity from capillaries and mitochondria. We get speed of contraction based upon this myosin ATPase speed. So people that are sprinters, they physically have an enzyme on their myosin that lets their myosin relax its contraction by using ATP faster than somebody that has more type 1s. And that's partially why they can run faster and they can jump higher, is that the machinery works faster, okay? It's the difference between having like a Ferrari and, I don't know, having my wonderful, like, old Subaru SUV that I have, okay? Some people are just race cars. Other people are, I don't know, more kind of economical in what it is that we're going around here, okay? Okay, Nia, your question.
Okay, so I have 2 questions.
Okay, fire away.
The type 1 muscles, so those are people that have like more endurance?
Generally, yes. Generally. You can have a lot of type 2 fibers, and if you run for distance a lot, you will turn those type 2 fibers into type 2As, and you can have a lot of mitochondria in them, but your myosin ATPase, this is where it gets complicated. Your myosin ATPase tends to be less efficient. It's kind of like a like a Hummer or something in the type 2s. And so your ability, you need a lot of mitochondria because you use a lot of ATP. But so you can be like a good middle distance runner or something, like an 800-meter runner, maybe even a miler. You need quite a bit of type 2 fibers because it requires so much like actual kind of raw speed in that particular way. But like people that are going to be good at, say, 10,000 meters or a half marathon or a marathon, that kind of distance, type 1 is what you really, really need for that, if that makes sense.
And for the myosin ATPase, dang it, I forgot my question.
Okay. When it comes to you, let me know. And
Okay.
we'll get back at it.
Okay.
So here we have just a flow chart or kind of a thing here that's going to talk about some of the differences between type 1 fibers, type 2A fibers, and type 2X fibers, okay? I could envision a scenario whereby this might make a reasonable question to ask about, say, compare and contrast the different muscle fiber types or something on your test. Like which ones are good for endurance and why? Which ones are good for sprinting and why? Or something that's going to kind of fall in that way, okay? As I mentioned, your fiber type distribution is determined by genetics. You can thank or curse your parents one way or the other for these kinds of things, okay? No matter how active you are, we can't change the percentage of your fibers that are type 1 or that are type 2. Within the type 2 fibers, because you have two different kind of flavors of them, A's and X's, if you are even moderately active, what happens to your type 2X muscle fiber types? They get turned into type 2A's. Type 2X fibers are so inefficient, they got like one good contraction in them, and then they're worn out and they're tired and they got to rest. So it's not very efficient evolutionarily to have a bunch of those. And so if you just walk, if you do resistance training, you're going to turn your 2X's into 2A's. But you can't turn 2A's into type 1. You can't turn type 1s into type 2s. There's no real way to get around all of that. People that have spinal cord injuries and are on bed rest for like years and years and years, if you're super, super inactive, you may get a little bit of conversion from type 1 to type 2 then, but that's a whole different kind of scenario and things that are going on, okay? So you got what you got, all right? And as I had mentioned to Nia, if you run or you do a bunch of endurance activities, you can make your 2A fibers have more mitochondria. You can get a lot better. It's a lot easier to get better at being a distance runner than it is at being a sprinter, right? We can work with your form and some things for sprinting, but you're either in some ways, you're either fast or you're not that fast. I can make any of you a lot better at running a 5K or a 10K than you are right now if you're just willing to come and work. You may never be world-class and elite at it, but you can tend to get a lot better. And that's because of the physiology that determines our performance on those kinds of things. Okay, so we talked about those things. All right, now we're going to move on to the nervous system side of things. We're going to talk a little bit about some structures that are going to be over there. Okay, so every time you want to do a contraction, okay, your brain sends a signal out through the brainstem, down the spinal cord, to some specialized neurons in the spinal cord that then send a signal out to that are then going to send a signal out to the muscles and tell them to contract. So if you're talking, if you're typing, if you're writing, I see Nia is chewing, whatever it is that you're doing, any kind of movement, the brain is the origin. The primary motor cortex is the origin of all of the signals that generate those movements, okay? Because skeletal muscle is voluntary. We're not going to get into brain anatomy. We're not going to get into, in many ways, kind of locations of where the primary motor cortex is. We're not going to talk about all of the secondary brain areas that have influence on all of this. I just want you to know it generally starts in the motor cortex, and it's going to run from the brain down the spinal cord, and then at certain locations in the spinal cord, it's going to interact with something called an alpha motor neuron, okay? Alpha motor neuron. In your spinal cord, there are these special nerve cells, and the alpha motor neuron is going to send a nerve or its axon from the spinal cord out, and it's going to make a connection at the neuromuscular junction to that particular skeletal muscle, and that allows your brain then to have this communication pathway where it can tell that muscle what to do. And all of this happens in just fractions of seconds. It's very, very fast, okay? Always blows my mind to think about all of those kinds of things, okay, that we can do all of that. So here's the basic structure of a motor neuron, okay? It has something called the cell body, which is this area right here. The cell body of the alpha motor neuron is located in the spinal cord, okay? That's what I want you guys to know. Connected to the cell body are going to be these little thin feather-like structures out here that are called dendrites. Dendrites are going to collect signals from up the spinal cord. So we've got, we'll just call this spinal cord that's coming down here, right? It's going to collect signals coming down the spinal cord with these dendrites. It's going to collect signals from, there are other neurons that are going to be sitting in the spinal cord around all of this that are called interneurons. It's going to get signals from them, and then that's going to tell the cell body what to do, basically. And so then when the cell body needs to talk to the muscle, it does something that we're going to call it depolarizes. When it depolarizes, it generates what's called an action potential. An action potential is an electrical signal, so it's electricity, okay? It gets signals, the neuron says, ooh, we need to depolarize because the brain wants us to make this muscle contract. It generates its action potential. That action potential then moves down the axon. So the axon is a long structure, a long tube that leaves the spinal cord. It's going to exit the spinal cord out through the foraminal space between your vertebrae, and then it's going to be in a nerve bundle, and it's eventually then going to move from the spinal cord all the way out down into the muscle that it's connected to. And then the axon begins to branch, and we're going to call these things axon terminals or axon branches and then terminals. And then each of these terminals is going to come into very close contact with individual muscle fibers or individual muscles. And that signal is going to get transmitted down and tell that muscle what to do, okay? Most of our axons that are going to connect to muscles have a fatty sheath that surrounds them that's made up of something called myelin. And then there are breaks in these myelin sheaths at places here that are called nodes of Ranvier. We're not going to get into all of the physiology of myelin sheaths and the nodes of Ranvier and the high density of sodium channels at nodes of Ranvier and things. But the point of having myelin and nodes of Ranvier is that because axons are so long, okay, imagine this. You have at say like S1 or S2, sort of down in your low, very, very low back, that's where the motor neurons that connect to the muscles that wiggle your toes are, okay? So they have to send an axon out of sort of your lower back all the way sort of across your butt, all the way down out to your foot, okay? And so for some of us, that's several feet to go down there, and it needs to be able to communicate very, very quickly because God forbid you step on something sharp or something super hot or super cold and you need to get your foot out of the way, okay? And so we need to be able to transmit a signal from the spinal cord out to those muscles as quickly as possible. And one of the ways that we're able to do that is by having myelin on those axons, okay? You have some nerves that don't have myelin, they transmit things slower. And there are some degenerative diseases whereby the myelin on the axons begins to degenerate and it goes away and it slows down transmission and it can cause some real problems, okay? So I just want you guys to just be aware of what some of those kinds of things are. Okay, so here's where things are going to get a little bit complicated, and I just want you guys to try to stay with me. I'll do my dead level best to make this as clear as possible, okay? In your spinal cord, okay, in your spinal cord, you're going to have the cell body of a motor neuron, okay? Each muscle in your body, so one muscle, okay, that one muscle has hundreds or thousands of motor neurons that will connect to that muscle, okay? So each individual, okay, but you've got way more fibers in a muscle than you have motor neurons in the spinal cord, okay? So you have, in some instances, millions or billions of, millions and millions of muscle fibers in a given muscle. And you may only have, you got 1 million fibers and you've only got 1,000 motor neurons. So there's not a 1-to-1 ratio with those things. So because of all of that, each motor neuron, its axon comes down and branches out, and it's going to branch and connect to some number of multiple muscle fibers in a given muscle, okay? The motor neuron and each fiber that it innervates, that group of fibers that it is going to connect to or innervate, that is called a motor unit, okay? It's called a motor unit. The motor unit is the functional unit of how the brain tells skeletal muscle to contract and generate force, okay? It's going to tell one motor neuron to fire and turn on those muscle fibers. And then if that doesn't get us enough force, then it's going to turn on sort of the next motor neuron and all of its fibers. And if that's not enough force, then we get the next one and the next one and the next one until we generate enough force to perform whatever movement we want, okay? The size of motor units is going to vary greatly, okay? You may have a motor unit that's very, very small that only connects to maybe 10 or 20 individual muscle fibers. And then in some of them, you're going to have motor units that are very, very large and connect to several thousand muscle fibers, okay? The size of motor units is going to vary along the lines of the muscle fiber type. And this is kind of where this gets to be a little bit confusing, okay? So let's imagine a scenario whereby in your muscle, you have both slow twitch fibers and fast twitch fibers, which most of us do, okay? The motor units that contain slow twitch muscle fibers tend to be small. The motor units that are going to contain type 2A fibers tend to be somewhere in the middle. And then the motor units that contain type 2X fibers tend to be the largest, okay? And so we will see in the next couple of slides that when you contract a muscle, it happens and it turns on motor units one at a time in a very orderly fashion. And it turns them on from small to large. So in every muscle during every contraction, you're going to start off by trying to turn on your type 1 fibers and then move to your type 2A fibers. And then if you need to, move to your type 2X fibers as we add in more and more motor units, okay? And so this is a unique property of kind of how the neuromuscular system functions and how it is organized, but it also is going to let us limit people getting tired and fatiguing because those type 1 fibers are going to get turned on first and they're very fatigue resistant. And it's also going to let us and allow us to very, very, very finely control the amount of force that we generate from a muscle, which lets us do things like write and talk and sing or play a musical instrument or throw darts or whatever those things are going to be that other, kind of other things we might not otherwise be able to do because of the way all of this is going to be organized, okay? So before we get into a little bit more of how all of this happens, I want to show you guys this, and I know you've seen this before, but motor neurons that all go to the same muscle are going to be located in a relatively similar place in your spinal cord, okay? And so if we look at these things, so like look at this, here we're going to have cervical vertebrae up here at the top. We've got our sacral vertebrae down here at the bottom. And I want you guys to note that, say, at roughly vertebrae C6 or C3 and C4, you're going to have the motor neurons that go to your diaphragm. And then at maybe C7, you've got tricep, okay? And then at C8, you're going to have things that go to your hands. We have very few skeletal muscles that are going to be in the thoracic vertebrae region. We've got a little bit that are going to be there. And then most of your lower body muscles are going to be down here in the lumbar spine or down kind of relatively high in the sacral spine in those ways, okay? And so by having all of the motor neurons that go to the same muscle in roughly the same location in the spinal cord, that's good because it helps us get kind of smooth contractions. So that, can you imagine a scenario whereby I had, let's say, some things that go to my quad muscles up here at C2, and then the rest of them are down here at, say, L3. And a signal from the brain has to move. So the brain is up here. The signal gets to here, and it tells these motor neurons to depolarize. They're going to start a contraction. Then it has to move all the way down here and tell the rest of them to start depolarizing. And you're going to get kind of out of time sequence on things, and it may be very difficult to get as much force as we want when we want it if those motor neurons were located in different places in the spinal cord. So by having them all in the same place, we can very easily turn all of them on nearly simultaneously, okay, compared to if they were spread out. That's good. The bad part is when you get damage to the spinal cord, let's say you had a spinal cord injury right here at T1, then everything below all of that is gone. You get no function below all of those things, okay? Whereas if you had some motor neurons that were above there that went to some of your leg muscles, maybe you'd have a little bit of preserved function, but we don't. So it's just kind of a trade-off in the midst of all of this, okay? All right. All right. So let's now talk about how am I doing on time? I'll do a little bit more here. Nia, you have a question?
I was on mute. So, okay, let's say somebody like had a spinal cord injury from like T10. So they would just be paralyzed from like their abdomen down.
Mm-hmm.
So how, because I watch a lot of like videos on like, you know, people getting paralyzed for some reason. But anyways, how do they still have like sexual function?
Oh, great question. So some sexual function, it's going to vary depending upon where their level of injury is, but some sexual function, kind of like bowel function and things, is controlled by the sympathetic and parasympathetic nervous system.
Mm-hmm.
And so they may be in a situation where, and I would need to look some of this up, not all of sympathetic and parasympathetic function. I'm trying to think about this. You need sympathetic function, like to get an erection, and I think you need parasympathetic function. Maybe it's backwards to get, maybe it's the other way around. Maybe it's you need parasympathetic to get an erection, and you need sympathetic function, like to ejaculate or something.
Mm-hmm.
That stuff doesn't have to come from the brain necessarily, right?
Mm-hmm.
And so if you can find ways to actually stimulate that portion of things, then even though potentially the person, like they couldn't feel it or anything, they might not actually experience that sensation in the way that it would normally be. You might actually be able to get, say, an erection or to be able to ejaculate, or, you know, in a woman, kind of the similar sort of processes in those things. So,
okay, so it can still function, it just, they won't feel anything.
Yeah, there's some potentially. I know there's some level of that stuff that can go on. And I would need to look up kind of exactly where the level of injuries are and things as to when people can and when people can't kind of have some of that stuff. Yeah, but you would very, very likely have experienced no sensation, but you could actually get the behavior or whatever, if you will, out of the whole thing.
Okay.
I was actually just in a class that talked a little bit about this too. And so there are some nerves in that region that like bypass parts of the spinal cord and will go way further up, or will just go straight to the brain. And so you can still get some degree of like sensation from those. It's not the same as they would have had prior to the spinal cord injury, but it's still like they can kind of tell what's happening. It's just like it feels different. But some of the nerves that can, like, depending on where their spinal cord injury, some of the nerves that bypass that area will still give them some degree of sensation.
Makes sense.
We did a lot of work in my doctoral degree. We had an NIH grant, and we did exercise in people with spinal cord injuries to try to prevent them from becoming type 2 diabetics. And so I should know, and I've forgotten, it's been 20 years, I've forgotten, you know, we had guys and girls. It's mostly guys that have spinal cord injuries because it comes from motorcycle accidents and, you know, other kind of dumb things for the most part. I know that we had several that, you know, they'd had kids, you know, kind of post their injury and those kinds of things. So I know that it can happen, Nia. It's going to be there. Okay.
Yeah, I have a friend that just had a baby and she's paralyzed like from waist down. So
Maybe good for having the baby. You don't need an epidural.
yeah.
So interesting. That of course now fascinates me on like how much sort of like involuntary contractions in the uterus and those things are versus, you know, trying to actively kind of like contract and push to get the baby out and those kinds of things. If you wanted to do it that way. Anyway, kind of off the subject. So let's talk about the place where the end of the axon or the axon terminal is going to make a connection to skeletal muscle. We call that region or that area a neuromuscular junction. And so at the end of an axon, where it's going to come into close contact, it does not physically make contact with the sarcolemma or the outside of the muscle fiber. And there's going to be multiple junctions per fiber, but where it does all of this, that's called the junction. Okay. And so it's a very highly specialized structure on both the nerve side here and the muscle side down here. Okay. So the axon is going to be kind of relatively thin. And then when it gets to right where it gets very, very near to the muscle, it's going to kind of form this bulb-like region that's going to go around like this. Okay. And so that's called a bouton, which you don't have to know. And then on the muscle end, there are going to be these folds in the muscle membrane, these folds in the sarcolemma here. And the purpose of both of those things is to increase the surface area on the nervous system side and on the muscle side. And we want to increase surface area so that the nerve has more area to release a neurotransmitter from these little vesicles in here. It's going to release something called acetylcholine. So we have, by having this kind of bulb region, we can have more acetylcholine. And then by having these folds on the muscle side, when the acetylcholine gets released into what's called the primary and secondary cleft here, you can have more surface area to put receptors. So these little green structures are going to be receptors for acetylcholine. And so having this specialized structure with lots of surface area is going to help us maximize our ability to capture the acetylcholine and get a good tight transmission of the signal from the nervous system into the muscle. Okay. So you get an action potential. It comes all the way down the motor neuron. It gets to the neuromuscular junction. So it goes from being an electrical signal. We're going to then release a neurotransmitter. It binds on its receptor. And then once that receptor is bound, then the muscle membrane is also then going to generate another action potential. And that action potential is going to move out in all these different directions on the sarcolemma. And then that's going to go down the T-tubule and into our sarcomeres. Okay. So there are some specific things that are kind of interesting about neuromuscular junctions. There are a lot of neurotoxins from spiders and snakes and things, or like the pufferfish, that are going to either lead to over kind of production of acetylcholine, or they're going to block the acetylcholine receptors. And so what happens is they either force you to have a muscle contraction and you can't relax, or you cannot begin to actually transmit. And so you can't generate a muscle contraction. And in either instance, most of the time, like if it's in people, if it gets into your diaphragm, then you will suffocate and you will die. And then there's some other kind of weird things, like it can cause paralysis and stuff, kind of out in kind of more peripheral muscles. Julia, you have a question?
Yes, I was just wondering how that works when you're having a muscular cramp.
So
yeah, that's it.
we don't fully understand the origin of all muscle cramps. There's probably some instances where you're getting some spontaneous release, let's just say, of acetylcholine at the junction from the nervous system side. And that may be due to some kind of weird unforced generation of action potentials down the axon or something. The most likely cause of the cramps, though, are going to be on the muscle side of things. And it's going to be kind of spontaneous release of calcium from the sarcoplasmic reticulum. Because you're like, I'm relaxed, I'm not doing anything, why am I getting any contraction, and those kinds of things. And so it probably is going to be somewhere distal even to the junction. It's going to be actually like at the T-tubule sarcoplasmic reticulum interaction would be my guess. But it's hard. We don't have a lot of data on cramps because it's somewhat ethically ambiguous for us to try to give people cramps and those kinds of things. So good question. Okay, so we've talked about what an action potential is already. We've talked about this. Okay, let me do this last little piece here, and then we're probably good. We can stop here for today and then finish up the rest of all of this tomorrow. Okay, so there's some other structural things. Julia, you have another question? No, okay.
No, I forgot to put the hand down. Sorry.
Oh, okay, no worries. So the other thing that we've not really talked about yet, okay? So you get your action potential, it moves along the muscle and it runs and it jumps down the T-tubule, okay? It depolarizes the T-tubule. The T-tubule connects to the sarcoplasmic reticulum at these places right here. Not for anything, there's a thing called the dihydropyridine receptor, which you don't have to know that is going to be the junction of those two things. But basically, action potential in the T-tubule tells the sarcoplasmic reticulum to open up and release its calcium out into the surrounding kind of area around those sarcomeres. So that calcium is going to be one of the first signals to turn on and make a muscle contract, okay? And so what happens is on the actin filaments, here's our actin filament here. On the actin filament, there are two different what are termed regulatory proteins. And those regulatory proteins, one of them is called troponin, the other one is called tropomyosin. And I do want you guys to know these two things, okay? So troponin is a kind of oval-shaped structure, and it's going to have multiple subunits. And one of those subunits, its sole function is to bind calcium. So the sarcoplasmic reticulum releases calcium. That calcium binds onto the C subunit of troponin, okay? That initiates a change in the shape of the troponin protein. It initiates a structural change. And so the troponin, it's like the little knob on your cell phone case that you twist that then turns your sound on and off or something. So it twists. As it twists, it's connected to this long, thin, rope-like blue structure here called tropomyosin. The best way for me to describe tropomyosin, all of my examples and analogies have to do with toddlers, but tropomyosin are the little plugs that you put, the little plastic plugs you put in your sockets, in the outlets at your house, so that when your daughter tries to jam her crayon or a fork or her finger or something else into it, that it can't shock her, okay? Tropomyosin is a long rope structure, and what it does is it lays over and covers up the binding site on actin for myosin, okay? I'll its some other, I'm not very hip, but myosin is very thirsty for actin, you guys. It really likes it some actin. It wants to touch it. It wants to grab onto it and hold onto it very, very tightly, okay? And so if we don't have tropomyosin, then myosin would all the time would be grabbed onto actin and it would be holding on to it, and you would exist in a perpetual state of cramping, okay? Just every muscle in your body, you'd just be like, I'm all cramped up all the time, okay? So normal resting state is relaxed. Tropomyosin covers the binding site. Myosin can't interact with actin. That's good, okay? Calcium gets released. It binds to troponin. Troponin twists, and as it twists, then it pulls the tropomyosin off the binding site, and now here comes actin, or here comes myosin, sorry. Myosin runs in, it grabs onto the actin molecule, then it does its power stroke and it pulls the actin, then it lets go and we start the whole process all over again. Okay. And so in this particular way, normal resting function of skeletal muscle is no contraction. It's relaxation. And so then when we want to have it turn on, we go brain, down the spinal cord, alpha motor neuron, down the axon, to the junction, release acetylcholine, action potential, down the T-tubule, release calcium, calcium onto troponin, move tropomyosin, boom, now we get actin and myosin together. Okay. And then we reverse all of that and shut it off right like that. Okay. Emma, question.
So are there like some forms of paralysis where the issue isn't with the spinal cord, but instead is with like some sort of like malformed troponin or something where like can't bind calcium? Is that a thing, or is that just a theoretical thing?
It could certainly be a theoretical thing. I am not well versed enough in all forms of paralysis to say for sure. One of the things, my guess is that like if you have a malformed troponin or tropomyosin, that you're not gonna survive as a fetus and those kinds of things. That's going to be such a deleterious mutation that you're not going to be able to do anything, because if it's malformed there, it may also be malformed in like cardiac muscle and things, and it's just not gonna, things aren't going to work correctly. But in practice, right, if I gave you a drug of some sort, let's just say, that was a competitive inhibitor and would bind onto troponin at the site where calcium binds, then I could very easily induce paralysis of you, right? Because then you couldn't turn on those muscles in that way, because there's no ability for calcium to actually get onto the troponin, right? And so most of the neurotoxins and things that I am aware of work more so on either blocking action potentials in the spinal cord or in the axon, or they actually block the release of acetylcholine, or they competitively inhibit acetylcholine at its receptor. So I'm trying to think of something that's called tetrodotoxin, which is in pufferfish, which is like a delicacy. If you go to get sushi and things, and if they prepare it wrong, it kills you because it competitively binds and blocks acetylcholine from binding, and you suffocate. Basically, you get paralysis and you can't breathe and those kinds of things. So that would be my guess. But that's an interesting thought exercise for sure, Emma, to think about it in that way. Okay, Nia.
Okay, so overall, like, what does troponin and tropomyosin do, like, in just, in conclusion?
In conclusion.
So I can write it down.
Okay. Troponin and tropomyosin are what we call regulatory proteins. They regulate the binding site on the actin proteins where myosin can bind to actin. Okay. And their functions are twofold. The function of tropomyosin is to stop myosin or prevent myosin from binding to actin, right? So its job is to make sure you don't get a muscle contraction. Whereas troponins is the opposite. Okay, the job of troponin is to, when it binds calcium, is to pull the tropomyosin away from the myosin binding site and allow myosin and actin to bind, and then cause or initiate a muscle contraction.
Okay, gotcha.
Cool. Okay, that's good for today, guys. We're going to stop here. Okay.